Hi, I am Krishnan, Assistant Professor, PG and Research Department of Microbiology, St.Joseph College of Arts and Science, Autonomous Cuddalore. Today, I am going to present the topic about histoplasmosis infection, which comes under the subject medical mycology. Histoplasmosis is said to be a systemic disease. Most of the infections can be seen in the reticuloendothelial system, and the pathogen name is Histoplasma capsulatum. This infection can also be occur apart from the reticuloendothelial system in the lungs, liver, and then spleen. In case of children, the infections are more frequently seen in liver as well as in the spleen, and it is called as hepatosplenomegaly. While in case of adults, the infections are primarily seen on the lung region, that is called pulmonary diseases. And this fungi normally reside within the reticuloendothelial cells, that is intracellularly. The pathogen Histoplasma capsulatum, it is said to be a black bird roost. So that is the origin of its living. This infection can typically spread through chicken manure around its gardener, and 3 weeks later will develop pulmonary infection. So hence it is said to be an occupational disease. So this infection is said to be a systemic mycosis. So this is a dimorphic fungi, and there are 5 major dimorphic fungi which will be causing systemic mycosis. So one is Histoplasma capsulatum, second is Blastomyces dermatitis, third Paracoccidioides brasilensis, fourth Coccidioides immitis, fifth Cryptococcus neoformans. 4 of these pathogens, which is a dimorphic, like capsulatum, dermatitis species, brasilensis species, and immitis, they grow like a filamentous mold in culture media. So when they infect the human being at the temperature of 37 degrees Celsius, they transform into yeast cell, that is unicellular stage. And this is the image shows of different fungi, both the mold form as well as yeast form. The second image, in the patient who is clinically infected with histoplasmosis, generally occur one of the 3 forms. So either it comes under acute pulmonary infection, or the next stage is called as chronic pulmonary infection, or the more serious form is called as a disseminated form. So there is generally a complete recovery from acute pulmonary form, where the infection can be like a flu-like diseases. However, if it is untreated, so that time the infection can spread throughout the body, that is called disseminated stage, so which is said to be very fatal. So one can able to notice some of the signs and symptoms of this infection, like shortness of breath and cough, which become protective. And if you see the sputum of the patient, which is said to be purulent and bloody, and the patient can also shows anorexic and weight loss. They also having night sweats. So these symptoms are once again the similar symptoms of tuberculosis. And also the X-ray report can also suggest the person is infected with the tuberculosis. But now the radiologist can able to distinguish, so this diseases based on the chest film. So this is the tuberculate conidia, that is called macroconidia and microconidia of Histoplasma capsulatum. So this is the yeast cell of this Histoplasma capsulatum, so which is phagocytosed inside the bone marrow. And this is the image of Giemsa strain. So comes to the pathogenesis of this infection. The infection occurs when the microconidia are inhaled by human being, and it is not transmitted from person to person. So this microconidia, when enter into the human body, it will be easily phagocytosed by some of the macrophages. Then inside the macrophages, they can multiply. Then through the macrophages, they can be disseminated to many organs and circulate throughout the body. So in acute stage, one can able to see the lesions in the lungs, and the type of lesion is called as lung calcification. It's nothing but the accumulation of calcium ions throughout the lung region. So this condition will be resemble like a tuberculosis. And most of the infections are spontaneously recovered. And some diseases, that is in disseminated stage, they require intense treatment and also a lot of antifungal therapies. So it comes to the lab diagnosis. The clinical specimens sent to the lab depend on the stage of the disease. The most frequent samples are sputum, then bronchoalveolar lavages if it is pulmonary disease, or if it is a biopsy material that can be for diseased organ. Bone marrow samples an excellent source of the fungus, so which tends to be grow in the reticuloendothelial system. And peripheral blood is also a source of visualizing the organism histologically. The yeast is usually found inside the monocytes, and many times as astute medical technologists performing a wild blood cell count will be the first one to make the diagnosis of histoplasmosis. And in some cases, the gastric washing are also a source of Histoplasma capsulatum. And people with pulmonary disease can easily diagnose it. And also sputum can also be used for the isolation of this fungi. This organism can also be confirmed by culturing. So this organism can be cultured in a media called Sabrodistus agar and incubated at room temperature. So this organism, it form a mycelium which is white, cottony in appearance. And the infection can also be diagnosed by serological method, where one can able to find the presence of antibodies or antigens in the serum sample. And the treatment for this infection, histoplasmosis, is we are using some antifungal drugs. The most effective antifungal agent for the treatment of this histoplasmosis or amphotericin B, then can also prefer ketoconazole and also itraconazole for a consecutive of 3 weeks. And when this organism will be growing in a Sabrodistus agar at the temperature of 25 degrees Celsius, as I said, the colony will be white and cottony after 2 to 3 weeks. And the mold phase of this Histoplasma capsulatum, it is characterized by thin branching septate hyphae that produce microconidia. And this will be entirely different from a tuberculate macroconidium. We can also confirm this infection or pathogen by using molecular probes, molecular DNA probes. in the blood sample. And this is the image, and this is the colony appearance. So as I said, the serological tests for this diagnosis, there are 4 important tests will be performed to confirm the infection. So one is called a lattice agglutination test, a complement fixation test, immunodiffusion, and ELISA. And this is the image of immunodiffusion of this organism, which can be shown like a precipitin band. So each of the serological tests has a different characteristics that make them useful. So one is called lattice agglutination test, a complement fixation test. So each and every test has its own benefit. So one can estimate the concentration of antigen or antibody in the sample also by using quantitative tests. So we can also go for immunological test called radioimmunoassay for the diagnosis of polysaccharide antigen. So as I said before, the preferred antifungal agent for this is amphotericin B, itraconazole, and voriconazole, which is effectively used. Thank you.